So guys, this is going to be a little bit of review to start off with, and it's also going to be a nice opportunity for us to sort of connect all the things we've talked about so far and kind of see how they interact with each other, which is really good. First of all, let's just do a little review of like what active transport means. Yell me some things that it means. We kind of talked about it on the check-in. Tell me something about it.
It requires energy.
Yeah, it requires energy. Just anything about active transport. So active transport requires energy. Now, we're going to tie some things together. So we're talking about cells here, using energy to move stuff. Where does that energy come from? What part of the cell? What organelle? Good, great. So I'm going to say this energy comes from the mitochondria. Okay, now we're going to take a step even further back, and we're going to think about where does the mitochondria get the ingredients to make the energy? What do you think?
Food we eat.
Yeah, exactly. Specifically, what foods?
Carbs and fats.
Carbs, mostly
Lipids.
carbs. Yeah, carbs and lipids, but for mitochondria, it uses carbs. So I'm going to say this is produced from sugars you ate. So this is why we've done things kind of in this order, because now we've built up to how we got the sugars, what used the sugars to make the energy, and now what the energy is used for. Now we've arrived here. In biology from now on, I'm going to use a different word to mean energy. It's 3 letters. Anyone know what it is? It's energy for your cells. It's a molecule called ATP. We'll talk more about that in our next unit, but from now on, just start like getting into your mind that energy and ATP is the same thing. Okay. Tell me another thing about active transport. What direction does it move stuff? From high to low concentration or low to high? Low to high. Good. So this moves things from low to high concentration, which we call with or against the gradient. Say it again.
Against?
Against, yeah, good. This is against gradient. So think about this like the gradient is a lazy river. And in active transport, it's like you're trying to go the opposite direction that the lazy river is pushing you. That's going to use energy because the current's pushing you the other way. So that's against the gradient. If it's passive, it's you in your inner tube floating down the river, having a great day. Passive, no energy. It's lazy, okay? All right, so now we're going to add on to this with something new. And it is that it also can move giant amounts of material or giant materials at one time. And that's what we're going to be talking about today. So active transport could just be moving things against their gradient, but it also could be moving like massive, 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 massive things. That's what we're going to talk about now. It's either going to move the massive things into the cell or out of the cell. Those are the only two options for this. And there's fancy words to describe both of those things that are actually not that complicated. You guys done with this so far? Can I go to there? Okay. All right, so the first thing we're going to talk about is moving huge things or huge amounts of things into the cell. And that is called endocytosis. If we break up this word, endo means in, cyto, what is cyto? Anyone want to guess? It's not. A good guess. Think about cell, yeah, because cytoskeleton is the skeleton of the cell. Cytoplasm is like the plasma of the cell, the jelly. So anytime you see cyto, it means cell. So literally this long word, endocytosis, all it means is into the cell. That's what's happening. So I'm going to describe these pictures first, and then I will write some stuff down. There's three types of endocytosis, but really we're only going to care about these two, phagocytosis and pinocytosis. What this is the cell eating is this one, and the cell drinking fluids is this one, if I'm going to make it like really simple. So in this one, I want you to transport yourself back to middle school for a moment when you learned about amoebas and like the different protists. Do you guys remember that a little bit? Maybe? Okay. So you might remember amoebas have these like fake feet called pseudopods that like branch out and wrap around the amoeba's food and like trap the food inside the amoeba's body. Yeah. Your cells can do that too, which might be like, what? But they can. So in this picture, this yellow line is the cell membrane. What's happening is the cell membrane grows these fake feet, these pseudopods, around a food source that's really big, like the size of bacteria, and then attaches to each other on the other sides, like right here. And then that pinches off and traps the food inside a vacuole, which is like a storage container to be digested. Now, organo review. This food vacuole is going to fuse together with a different organelle that will digest it. What is it?
Lysosome?
Yes, it's a lysosome. Because lysosomes' jobs are to break down either waste or food or damaged cell parts. And they contain these enzymes that will do that. So this will fuse together with a lysosome, and then the lysosome will break it up into tiny pieces that your cell can then use for whatever it needs to use them for. That's phagocytosis. Pinocytosis is very similar, except instead of growing out, the cell dips inwards and just allows some of the fluid that's already outside the cell to like suck into the pit that it creates, and then just pinches off and forms a vesicle, which is a smaller vacuole. It's just a tiny vacuole. And that is, now there's fluid from outside inside the cell that the cell can use for whatever it needs to use it for. The last one, which I'm just going to talk about, but I'm not going to write about, is this one, receptor mediated, which would only be used if the cell really needs one specific type of molecule, like a hormone. So what will happen is the cell has these special receptors that only attach to the star molecules. And when the star molecules attach, it triggers this pit to start to form, just like it did here, which results in only the stars being inside the cell. So it would be like a specific hormone that was a signal, or a specific food type that needed to be absorbed by the cell. It could use receptor mediated. If you ever take AP Bio, you'll learn more about that one, but we're not learning about it in here. Okay, so let me zoom over here and tell you about phagocytosis and why we care. So phagocytosis is the cell engulfing giant solid particles. Now, here's why we care about phagocytosis, is that your immune system actually has cells that are called phagocytes, which means it's their only job to go around and eat stuff. And what they're eating is bacteria. So there's a picture of one. That's what it looks like. It goes up to the bacteria, eats the bacteria, digests the bacteria, and then what actually happens when they've eaten a lot of bacteria is they die and they explode. And then what happens is your snot turns green because of all the like dead cell parts that are in your mucus. And then you know that you're sick because your immune system is working. Ta-da! Yeah, Toby.
What is pango mean?
It means like eat. Yeah. But that's amazing. And these things inside these white blood cells are basically like giant lysosomes. They just contain like tons and tons of enzymes that break up the bacteria and make sure that they don't infect you. And then they die so that nothing bad can happen. And then you sneeze them out. Okay, so I'll say example is immune cells consume and break down bacteria. That's not how you spell that. To prevent infections. And then the other type is pinocytosis. Which I'm not going to do an example of, but that is just the cell taking in large amounts of liquid. So we call this like cell drinking. And this one is like cell eating. And both of these together are types of endocytosis. So they both would be considered active transport, things going into the cell. Anyone have questions so far? Okay, cool. So then the only other thing we have to talk about related to this is the opposite, which is exocytosis, exiting. Do you guys need more time? A little bit. All right, so now there's one other picture on here, which is down here. And this is exocytosis. And I chose a picture that was a little more zoomed in so you could see what's actually happening like with the membrane. I'll just label this as exocytosis. And just like we did with endo, exo means out. So think about like exit, leave. Just like I did with endocytosis, I'm going to describe this first and then I will write about it. So here's what's happening is you have this vesicle. And for this example, I'm going to talk about, we're going to say that these like purple dots are proteins. And we're going to say that this is a pancreas cell and that the proteins are insulin. Insulin is really important for regulating what? Do you guys know? Blood sugar. Yeah. So that means if the insulin is inside the pancreas cell, can it do its job? No, because it needs to be in the blood to regulate blood sugar. So after the cell produces the insulin, which is a protein, so which cell part made it? Which cell part makes proteins? Good. So the ribosomes produced this insulin, and then it sent the insulin to another organelle to pack it up and put it in this neat little vesicle. What packs it up? Yeah, exactly, Golgi's. So it's now been made by the ribosome, sent to the Golgi body, gotten packed in this neat little membrane vesicle, and shipped out towards the cell membrane. So here it goes, arrives at the cell membrane. Literally, the cell membrane and the vesicle membrane are made of the same thing. They're both made of phospholipids. So what happens is the vesicle fuses together with the cell membrane, and that allows it to open up and release the insulin into the blood. Now it can do its job, which is really important. So kind of another cool thing about this is that all the time, the membranes of your cells are being recycled and moved around. So this membrane is becoming part of the cell membrane, but then when something else is received, the membrane will pinch back off and become another vesicle or vacuole, right? And then also the vesicles can like travel to other cells. And so the cell membrane of one of your cells might later be used for a different cell. And it's all the time being recycled and reused and moved around, which is just like kind of cool. But all the membrane in your body is the same and can be incorporated to any other cell type, which is cool. So if I'm going to describe this, I'm going to say materials are, I'll say, exported from cell. And I'll say my example is insulin produced in pancreas cells is shipped out into blood. So these abilities to transport large amounts of things are really important for keeping your cells functioning, whether they're eating or they're releasing products they made, or maybe they're releasing waste. We didn't really talk about that, but they could be. Okay, so when you're done with this, the only other whole group thing I want to do today is make like kind of a cheat sheet chart for you guys about the different transport types that you'll be able to use for your test. So when you're ready to do that, I'm going to make a new page in OneNote. So if you just come down to the bottom and press add page. Are you guys done with that? Yeah. Okay. So press add page, and we're going to call this transport types sheet. And I'm going to put lines on mine. So I'm going to go to view, rule lines. You don't have to put lines on yours if you don't want to, but I like them.
What do you go on there?
View at the top, and then press rule lines. And you can choose your like thickness. Okay, and then in my little chart, I'm just gonna make it, I'm not gonna do lines because lines stress me out. So I'm gonna do transport type. And then I'm gonna do active / passive. And then I'm gonna do what moves. And then I'm gonna do where. And we're gonna start with our simplest one, which is diffusion. Okay. So we talked about this in our check-in, but diffusion, active or passive? Passive, right? No energy needed. So this is passive. And then we're going to think about what type of things are moving. So are they going to be big things or small things? And are they going to be non-polar things or polar things? Smaller things, right? So small. And then what do you think, polar or non-polar? Toby?
Polar.
No. Why not? Why non-polar? Because what is the membrane made of in the middle? Lipids, right. And lipids are non-polar and they repel water. So we want things that are similar to the lipids to go through the membrane. So they have to be non-polar. So say small, non-polar molecules. And those are the only things that are going to be able to pass. And then where are they going? From high concentration to low or from low concentration to high? High to low. Good. So they're going from high to low concentration. Which are we going to call that with the gradient or against? With. With gradient. They're floating down the lazy river. All right. From there, we're going to go to facilitated diffusion. Facilitated diffusion is just a type of diffusion. So is it going to be active or passive? Passive. Okay, so now Toby, what's going to move? This one will be polar, right? So this is going to be bigger. I'm not saying big because not as big as like endo and exocytosis, but bigger. Polar or charged molecules. So things like salts, sugars, ions, they're going to need to do facilitated diffusion. And then same thing, this is going to be high to low. And I'm not going to write the gradient thing, but it's still true. It's still the same. The next one is osmosis. Osmosis is what your lab focuses on. So, active or passive? No guesses?
Both? Or
Well,
either?
it could be, but to keep things like simple, we're gonna say passive. Active osmosis is possible, but we're gonna loop that under active transport. Okay, guys, in osmosis, only one thing can move. What is it? Osmosis is the
Water?
water, right? Diffusion of water. So water only. And in osmosis, it's passive, so it's moving from high to low. And I'm going to say like star of water, which remember matters because where there's more water, there's less salt. And where there's less salt, there's more water. So it's important to know that it's moving towards the lower concentration of water, not the lower concentration of salt. Okay. All right, we're going to loop active transport all together. So active transport obviously is active, it's in the name. Any molecule could do this.
At this time, all first lunch students have 5 minutes to get to their next class.
The only thing that makes it active is the direction it's moving. So it's going to move from where to where? Low to high. Exactly, low to high. So it's going to move from low to high, which is against our gradient. So like I was just telling Liam, water can do this. If water is moving to where there's more water, it would be active. Active osmosis, but you could just call it active transport. And then the last one, we're going to stick endo and exocytosis together. So endo / exo cytosis, we just said was active. And this is going to be giant things or big groups of things. And since it's active, we're going to say that it's low to high. And those are all the transport types that you will need to know for this class. When you finish with this, the only other thing we're working on today is also in OneNote. It's called transport types practice, or like cellular transport types practice or something like that. Should be right in there with these other pages.